Good day, ma'am. Our lesson entitled Midterm Laboratory Exercise. Before we proceed to our lesson, let's start with the lesson objectives. Students should be able to explain the importance of semen analysis, analyze the forensic examination of semen, and these are the content. Title 1. Title objective. Slides, contents, and presenter's identity. Number 2. Description of semen, sperm, and semen analysis. Number 3. Difference between presumptive for semen and confirmatory test for semen. Number 4. Step in collecting DNA evidence for semen. Number 5. Importance of semen analysis in criminal investigation. Now, I will discuss the semen, sperms, and semen analysis. Let's start with semen, and it's the fluid released through the penis during orgasm. Semen made up of fluid and sperms. The fluid come from prostate, seminal vesicles, and other sex glands. Next is the sperm. It is the male reproductive cell or gamete in isogamous forms of sexual reproduction. And the last, semen analysis. It's the great value in medical legal practice like alleged rape cases or sexual assault. The primary aim of forensic semen analysis is to sample and example smears or other biological material taken from the assailant, the victim, or stains found on clothes and other evidences. And there are three presumptive tests for semen. Then the first one is visual and alternate lights. Source semen can be detected with the naked eye, in clothing and other garments, or through the use of ALS such as ultraviolet light. And the second is acid phosphatase test. Using standard chemical reaction, important laboratories and analyze a given stain for the presence of these enzymes. In the presence of contained stain, AP will produce a dark purple color in less than a minute. Last, the prostate specific antigen. It is also known as PSA or P30, can be detected in semen. PSA is produced in high amount by the male prostate glands but can also be found in very small amounts in fecal matter and breast milk. And now, there are two confirmatory tests for semen. First, Christmas tree strain. It is a positive identification of sperm cell. The first stain, the neck and tail of the sperm. Cell in the green and blue, whereas the acrosomal cup with red and pink. Next is the RSID. Semen strip test. This test provides sensitivity and specificity to human semen, similar to the pregnancy test strip. RSID semen test identifies the presence of the seminal beside specific antigen. And for now, steps in collecting DNA evidence. First step, absorb suspected liquid semen on a clean cotton swab and air dry the swab and pack in a clean envelope with sealed corners and clean corners. Second step, submit small and dry suspected semen struck object to the laboratory, pack in clean envelope with sealed corners to protect them from strain removal. Third step, documentary scene. Develop written documentation, photograph, locate and interview victims and witnesses. And the fourth step, processing evidence of the scene. Organize evidence, processing and control scene contamination. And the last step, completing and recording the scene. Make sure all investigative steps are documented, release the scene, and submit the report. And last, for the last slide, the importance of semen analysis in criminal investigation. Semen analysis is very important in assault cases as it's very useful for detection of semen in clothes, isolation, and identification of seminal stain found on variants suspected such as clothes, bedsheet, pillows, blank sheet. Analysis of various protein present in semen or sexual stain. Detection of sperm, cell in vagina and other body part, and facial matter examination. And the presence of semen can be important to a crime scene investigation. The visual detection of semen can provide evidence in genetic material present in the sperm can be analyzed and used as a genetic fingerprint to identify as suspect. And that would be all for the midterm laboratory exercise titled Semen Analysis. Thank you.